Okay, so this is chapter 10, part 2, and there's only 2 parts to this chapter, so this is the last section. So here we're going to dive into the cell cycle and mitosis, cell division. So to start off with, let me give you an overview, general definition of the cell cycle of mitosis. And so mitosis is going to be cell division, in which we go from one parent cell to 2 new cells, which we're going to call those cells daughter cells. And those daughter cells are going to be genetically identical to each other and genetically identical to the parent cell. And so before we get into the stages here of the cell cycle, I want to call your attention to what we call G0. And so you can see this is kind of, it comes out of the cell cycle. And sometimes it can go back into the cell cycle. So you can see that arrow going back in. So what is this? So there are going to be some cells in an organism's body, including humans, that are not going to divide very often or not going to divide at all. Some cells will, not divide for a long time and then will be triggered to do so. So again, they can go into G0 and some cells can come out of G0 and re-enter the cell cycle. So a cell in G0 we would consider a non-dividing cell, and we also use the term quiescent. And so a cell in this quiescent stage, again, it just means non-dividing. The cell is still active, it's still living, it's still doing its normal daily functions, it's still doing its job, it's just not dividing. Okay, an example of, cells inside of us that tend to go into this G0 are cells like our neurons. Okay, they're going to not really divide very often, if at all. And again, some cells will stay in G0 and some will re-enter the cell cycle. They're triggered to re-enter the cell cycle. Back up one slide there. Okay, so let me give you just a brief overview now of the cell cycle. So there's really 2 major phases here. There's interphase and then there's the mitotic phase. And so if we go to the center of our cell cycle picture here, we can see in blue, the larger phase is interphase. And then in yellow, we can see this M, which stands for the mitotic phase. And so we will go over the subphases of each. So interphase is broken up into G1, S, and G2. And the mitotic phase is broken up to prophase, metaphase, anaphase, and telophase. And at the end of telophase really is cytokinesis. Okay, we're going to go over each of these stages individually. But before we do, I'll just introduce you to the basics here. So again, interphase shown in blue in the center of the circle, the cycle. Cells that are in interphase are going to be doing normal growth, and they are going to be preparing for cell division. And most importantly, during S phase of interphase, bolded here for you, you can see how important this is DNA replication. So all of the genetic material of the cell gets copied. So we go from one complete set of the genome to 2. Those 2 sets of DNA will then be divided during the mitotic phase. So again, shown here in yellow. So the replicated DNA and the rest of the cell, all the other organelles, the cytoplasm, they get split up and divided into 2 daughter cells by the end of mitosis. Okay. So interphase, let's jump in and talk about this in a little bit more detail. So again, interphase is going to come before mitosis, and you can see as far as length, so if this is divided based on, kind of length of the stage, interphase is long, much longer than the mitotic phase. Again, what's happening here, normal growth, getting ready for division. We got G1, S, and G2. So let me give you some specifics on each of these sub-phases. So G1, unfortunately, the G actually stands for gap, which I kind of hate. When I think of gap, I think of like this resting cell not doing much, and that's really not true. So even though it stands for gap. I kind of think of growth. So G1, again, my mind goes to growth, even though it's supposed to represent gap. What happens here? So if you were to look at a G1 phase cell, you really wouldn't see a lot of change. However, the cell is very active biochemically, and what it's doing is it's preparing everything that's needed for DNA replication. Okay, and it's also, starting to package chromosomes a little bit, getting them ready to eventually really compact once we get into the mitotic phase. Okay, so stuff is happening again during G1. The cell is preparing for DNA replication. During S phase, that S stands for synthesis. I like that. That's good. So what's being synthesized? DNA. So during S phase of interphase, all of the DNA will be copied. Okay, it's replicated. And if we were to think about this in terms of chromosome structure, we would say that chromosomes go from the monad form to the dyad form. And so let me just draw this for you. So even though the DNA is not compacted yet, I'm going to draw it compacted just to show you. So this would be a monad chromosome, one arm, and this is a dyad chromosome, so two arms. Okay, and so what happens during S phase of interphase is the DNA here of this monad gets replicated, and so these two arms, the product of S phase, are exactly completely identical to each other. Okay, so this is what we get after DNA replication. And so again, your DNA is not condensed yet at this point, but if it were to be condensed, this is what it would look like. So this is a monad chromosome, one arm, and this is a dyad chromosome, two arms that are genetically identical to each other. We can say that these two arms are sister chromatids. So again, on these notes, I would go ahead and make some sketches for myself to make sure that I knew what these terms mean. So again, these are sister chromatids, 100% identical to each other. They are going to be joined here in this region by proteins, which are going to make up what's called the centromere. Okay. So again, during S phase, we go from monads to dyads. And those dyad chromosomes, again, are made up of two identical arms, which are called sister chromatids, that are joined by the centromere. Okay, so we say chromosomes are in the dyad condition. What else happens? These structures called centrosomes also get duplicated. So again, let me take my pen back out. So if this were my interphase cell, and I will, this is not a great picture, but there's an actual picture coming up. But if this is my interphase cell, my nucleus would be intact. And I would have nucleoli, some of these dark stained regions. And there's going to be DNA in here, again, that's getting replicated. And it's not going to be really tightly condensed. It's going to be in the form of chromatin, which is DNA with associated histone proteins. But again, not condensed yet. If I were to condense it, by the end of S phase, I would have dyads. Okay. So lastly, the centrosome gets duplicated. So there's going to be this structure that's made up of two elements of the cytoskeleton made up of two centrioles, if you remember that from chapter 4. And coming out of the centrioles are microtubules. So again, you might want to make yourself a sketch with some labels. Pen's a little slow here. So this structure during interphase will duplicate. So you end up with two of these. Right now I just have one drawn. Okay, by the end of S phase, you'll have two of these structures. What is this structure involved in? We're going to see in a few minutes, but this is going to make up what's called the mitotic spindle, which is responsible for pulling chromosomes apart during M phase, so during the mitotic phase, during mitosis. Okay. G2, the second gap, which again, I hate the term gap because it's not really resting. The cell's doing stuff. So what is it doing? It's preparing now for the actual division of the cell. So it's preparing for mitosis. It's making more energy, right? Because energy is going to be required for this process. And so the mitochondria are all ramped up, making ATP. Any organelles that have to be divided up, so ribosomes and the Golgi and the ER, all those things, more of them get made so that each daughter cell can have an equal amount. And lastly, elements of the cytoskeleton are going to break down. So the cell's going to totally devote everything it's got to replicating, dividing. And so it takes down most of the cytoskeleton. Okay, so that's interphase. Again, the most important thing there is that the DNA gets replicated. So we go into the mitotic phase, shown here in yellow. Okay, so what's going to happen? So first off, in mitosis, which is also called karyokinesis, sometimes you might hear that term, what happens here is the nucleus divides. So we go from one nucleus to two. And then at the very end here of telophase, we have another phase, which is really part of this, called cytokinesis. And in cytokinesis, the rest of the cell gets divided. Okay, so in mitosis, we really call that a nuclear division, and cytokinesis is when the rest of the cell divides. By the end of cytokinesis, so by the end of the mitotic phase, we end up with two individual cells. We call those daughter cells, and again, they're identical to each other and to the parent. They have the same DNA. They're pretty much clones. Okay, so let's go through these stages. So mitosis first. So mitosis, which again is also called karyokinesis, can be broken up into prophase, metaphase, anaphase, and telophase. In some textbooks, including the one that we use as a reference, so the OpenStax biology book, also throws pro-metaphase in there, which is really part of prophase. So you can think of it, as being kind of a sub-phase of prophase. So let's go through each of these in detail. Okay, so we've left interphase, okay? DNA has all been duplicated, but it's still in the nucleus, and it's still in the form of chromatin, okay? Centrosomes have been replicated, and now we're going to get into the actual cell division. So the first phase of mitosis is called prophase, so that's what that P stands for. This is the longest phase of mitosis. There's a lot of things that happen here. And so let's look at this list. So first off, the nuclear envelope, that double membrane structure that's surrounding the nucleus, it's going to start to break down. And so you can see in this cartoon drawing, there's like spaces in between. That's showing the beginning of this fading away, breaking down. What else happens? The nucleolus, that dark stained dense region in the nucleus where ribosomes are assembled, that disappears. So again, in this picture, we don't see it anymore. Those centrosomes that had been replicated, they're now going to move towards opposite sides of the cell, which we're going to call the poles. And in animals, again, at the heart of those centrosomes will be centrioles. Plants lack those. We don't really know why. Okay, so here we go. Here's one, and here's the second, and they're going to set up shop. They're going to separate themselves and go to opposite sides or poles of the cell. The microtubules start to form the mitotic spindle. So again, growing out of the centrosome will be these structures which are made of microtubules. Those are eventually going to attach to chromosomes and pull them apart. What else? Dyad chromosomes tightly condense. Okay, so during prophase now we take that replicated DNA and now we form chromosomes. And so you can see the chromosomes, there's 1, 2, 3, 4 here. And they're all in the dyad form. They all have 2 arms, or we could say 2 sister chromatids joined at the centromere. Okay, so again, this is a cartoon drawing showing that. And then we also have an electron micrograph showing that as well. Okay, notice in the cartoon drawing that there's more ER that's been made. There's multiple mitochondria that have been made. So the cell's, getting ready to really divide everything up, not just the DNA. Okay, and then here's this prometaphase, which again, some people consider this still part of prophase, but it's a subphase. So again, the events from prophase continue. A structure called the kinetochore forms at the centromere of each chromosome, and the mitotic spindle binds to that. Okay, so the nucleus completely fades, chromosomes completely condense, those centrosomes move completely to two different sides of the cell, and lastly, each chromosome attaches to the mitotic spindle. So you can see the spindle fibers attaching to the chromosomes. Okay, so if I were to take a dyad chromosome again and just that by itself, the two arms are called sisters. They're identical. They're joined at a structure called the centromere, and at this structure will be proteins that are part of what we call the kinetochore, and it's that structure that binds to the microtubules. Okay. Okay. The next phase is metaphase. And so when you think metaphase, I want you to think middle. And so during metaphase, really simply, the dyad chromosomes are going to line up single file in the middle of the cell. And the middle of the cell has multiple names. It can be called the metaphase plate, or it can be called the equatorial plane, or just plain old the equator. The middle of the cell, the midline. Again, depending on what book you're reading, scientists use multiple terms. In any event, chromosomes are lined up, single file, middle of the cell. Each chromosome on each side, so each sister, is attached to the mitotic spindle. So if you look at just this first chromosome, it's attached on both its left side of the kinetochore and the right side of the kinetochore. Sister chromatids are still attached to each other, right? The centromere is still holding them together. Okay, so after metaphase comes anaphase. And so when I think of anaphase, I think away. And so what's going to happen here is the monad chromosomes, or we can say the sister chromatids, will be pulled apart and they'll be pulled away from each other and away from that midline. Okay, so we can see that in the picture here, right? These chromosomes were just together and lined up in the middle, and now they're being pulled apart towards opposite poles or opposite sides of the cell. So in order for that to happen, the proteins that are holding the sister chromatids together, which are called cohesin proteins, they're going to degrade. They're going to be broken apart. And so the sister chromatids are allowed to come apart. The sister chromatids, which we can also call, in this case, monad chromosomes. So we go from a dyad in metaphase to now monads in anaphase. Monad chromosomes are pulled apart. So half of that original chromosome, or one chromatid, or a monad, will be pulled in this direction, and the other half, again the monad, one arm of that chromosome, will be pulled in this direction. So they're pulled towards opposite sides of the cell. So let's think about this. These two arms, right, if I were to put them back together, let's see if I can get my pen out here. So if I were to draw this like this, how it just was in metaphase, these two arms are genetically identical to each other, right? They were replicated or copied during S phase of interphase. And so now we are breaking them right here and pulling them in opposite directions. So this one's going to go to this side, and the other arm will go to that side. And so what we're doing is we are separating the genetic contents. We are separating the identical arms of this chromosome. And so over here, the genetic material over here is the same as the genetic material that's now being pulled in the opposite direction. Okay. Okay, trying to get out of the pen. There we go. Okay, so that's anaphase. So they're being pulled away. One more thing. So what are they being pulled by? The mitotic spindle. So shown here in blue, right? This whole thing is called the centrosome. And if this is an animal cell, these are centromeres in the center. And growing out of there are microtubules, which are part of the cytoskeleton. In this case, what are they doing? They're grabbing onto each arm of that original chromosome, and they are responsible for physically pulling the chromosome toward the pole. So the mitotic spindle is what moves this chromosome. Okay? Okay. Lastly is telophase. So telophase is pretty much the opposite of prophase. So what's going to happen here? So those chromosomes that were just being pulled apart, they reach the poles. Again, they're still in that monad or one chromatid form. They will then start to decondense. So they start to unwind and uncoil, and the nuclear envelope will reform around that genetic material. And so we're going to end up here with two nuclei being formed. The mitotic spindle, responsible for separating that DNA, will also start to break down. And so we can see here, the cell's not separated yet, but you can see, this division mark where we have one nucleus here. That genetic material is now in there and it's starting to decondense. And the same thing's happening over here. That nuclear envelope is reforming around this nucleus. The mitotic spindle has started to break down. Okay. And so again, by the end of telophase, we have two daughter nuclei. They're identical to each other. And lastly, we have cytokinesis. So when you think of cytokinesis, this is where the rest of the cell gets divided. And I actually missed a slide. Let me just go back. I like this slide a little bit better. Okay, yeah. So cytokinesis. So the DNA has been separated. We have two nuclei. Now we're going to separate the rest of the cytoplasmic contents and the cytoplasm itself. And so here we go. We've got two nuclei, right? And so we're going to put some of the elements, like here's the ER and Golgi, mitochondria. It's got its own centrosome here. And the same structures will be found on this side of the cell. And what will happen here is the membrane will eventually pinch off. And so you eventually will have two cells. Okay, and again, those two cells will be identical to each other. Now, how this happens in animal and plant cells is different. And so the next slide shows you that. So here's the difference. So in animal cells that do not have a cell wall, what will happen is something called the contractile ring will form. And this is made up of actin filaments. So the cytoskeleton is going to move to the center of the cell, and it's going to form a structure called the cleavage furrow. And in the cleavage furrow, really what's happening is these elements are going to pull on the membrane until it eventually pinches apart, and you end up with two cells. Now in plants, because of the cell wall, it's a little bit different. So instead of pinching the two cells apart, what happens is vesicles from the Golgi carrying cell wall components will move to the center of the cell and will start to lay down a new cell wall. In the beginning, when that first starts to form, it's called the cell plate. And eventually it continues to be formed until we have two distinct cells. So in animal cells, there's a structure known as the cleavage furrow that's formed. And in plant cells, we have a structure known as the cell plate. Those are key indicators of a cell in late telophase / cytokinesis. Cytokinesis and telophase, really happen kind of together. That's the end of chapter 10. So again, at the end of mitosis, what we end up with, whether we're talking about plants or animals, is we end up with two cells. Okay, and those two cells are going to be called daughter cells, and they're going to be genetically identical to each other and to the parent.